Welcome to the Brain and Behavior Research Foundation Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. James McPartland will present Developing Biological Markers to Improve Care in Autism. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 194 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the Foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has had such great impact is because we do not limit our focus to one condition. Since 1987, the Foundation has awarded $450 million to fund more than 6,500 research grants around the world, covering a broad range of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. James McPartland. Dr. McPartland is the Harris Professor of Child Psychiatry and Psychology at the Yale Study Center. He was a 2013 Claremont Prize winner and a 2009 Young Investigator, both with BBRF. Our webinar will begin with Dr. McPartland's presentation, which will then be followed by Q&A. To submit your questions, please use the Questions tab on the control panel on your screen. Please feel free to submit them at any time. Following the presentation, I'll ask as many as possible in the time allotted. And now, I'm pleased to introduce Dr. McPartland. Jim, the floor is yours.
Thank you so much, Dr. Borenstein. It's really my pleasure to be here. It's wonderful to have the opportunity to talk to you all about some of the work that I've been involved with. My plan today is to talk to you about work that we've done in the laboratory and in a consortium that I'm a part of, really to try to move autism clinical care to a place where we can utilize biology, which is a place that we are not quite at yet. Before I start, I just want to acknowledge the sources of support for the research in my lab, and those that support myself as well. I don't think there's any conflicts of interest with the work that I'll be discussing today. I do want to highlight one source of research funding in particular. As Dr. Borenstein mentioned, I was the recipient of the BBRF Young Investigator Award very early in my career. As I'll talk about today, I am a clinical psychologist who works directly with people with autism, kids and adults, and I'm also a researcher. And really, I quite literally have the opportunity to be a researcher because of the support from the BBRF very early in my career. So it really is, it's a wonderful experience for me to have the chance to come and present in this context, because much of the work that I will talk about today is derivative of the support that I received from the foundation. So let me give you an overview of what I would like to speak about today. So I want to start by talking very briefly about what autism is and why I think we need biological markers. When I say biological markers, I'm going to use the term today, biomarker, and I'm going to define that term for you and tell you what it is. I'm going to tell you why and how I think biomarkers could be useful in improving care. And I'll talk a little bit about the science of biomarkers. What makes a good biomarker? What makes a good biomarker specifically for autism? When we think about developing them, what does progress look like? And I'll tell you, some examples of work that we've done in a lab on one particular brain-based biomarker to advance it. And then really I'll also think about what the ultimate goal is. Where does this path lead, and how could we get to a place, in our society where we can use some of these tools to improve the experience of autistic people. And really, that's one big long road. And I also want to end by talking about some things that are just beginning, roads that are just beginning in our laboratory. One is whether these biomarkers can help us actually develop new approaches to treat, and also how we can be more inclusive of the full range of people on the autism spectrum, which we're not doing an adequate job of at present. That's the roadmap. Let's get started. So talking about autism, many of you know a great deal about autism. Some of you might not know much at all. Autism is a developmental condition. It impacts many different aspects of a person's functioning. What I've listed here are really those that are most well-defined and most central to the diagnostic taxonomies that we currently use. In the United States, the Diagnostic and Statistical Manual, the DSM, in Europe, the ICD. And really, when we think about all the different places that autism touches, the ones that are most important in diagnostic assessment are social communicative function, and you're going to hear me talk a lot about that today, interests and behavioral flexibility. So, and to give you examples, by social communicative function, this could be interest and comfort and agility in interacting with others, using eye contact, understanding eye contact, using other forms of verbal and nonverbal behavior, as well as conceptual things like insight into social relationships and connection to others. A second category is interests and behavioral flexibility, and this can be being interested in very particular things or unusual things, having insistence on certain kinds of behaviors, or even repetitive kinds of movements, stereotype mannerisms, for example. And then the third thing, which is actually a part of the same category, but I think is a little bit conceptually distinct, is response to sensory information, perhaps being oversensitive or undersensitive to sensory information. So it might be intuitive when I describe all these different kinds of behaviors and functions being relevant to autism, that autism is a really complex condition that's characterized by great diversity. And when we think about it biologically, autism isn't one thing. There are many different causes of autism. And when we think, especially the things that I think about neural systems in the brain, there are many, many different systems involved, many, many different mechanisms. So there's a ton of diversity, and that's a challenge for thinking about how to harness biology. You may have heard the expression, if you've seen one person with autism, you've seen one person with autism. And that expression is meant to characterize this diversity that we see. But I also think there's really some striking commonalities. And really, as a clinician, as a diagnostician, I can tell you that there is something that is true of every person who receives a diagnosis of autism. And that is that they have differences in the way they engage with other people. And for that reason, I really think even if we're not going to find a biological cause of autism, a biological mechanism in the brain, if we're going to try to find things that are meaningful to as many people with autism as we can, I think that looking in this social communicative area is where we might get the most traction. So I'm talking today about biology, but really where we stand at autism is reflected in the kinds of things that I've talked about. When I describe the things we use to diagnose autism, I didn't mention a single aspect of biology, and that is the truth. Right now, presently, we are entirely reliant on behavior. The behavior that a clinician like me sees when I'm working with a patient, the behavior that a parent tells me or tells another clinician that they've observed in a person at home. And so right now, we stand at a field in autism that we recognize it to be a biological condition, but in making diagnoses, in making treatment decisions, in all aspects of clinical care presently, we're entirely reliant on behavior. And I see that as a shortcoming. I think that we as clinicians have come very, very far, but I think that we are at a place where we could do so much more if we understood more about biology. And that's the imperative driving the work that I'll talk about today, is how can we get understandings of biology into the clinical settings. And I just want to drive this home with a really concrete example. So as a scientist, research studies in my lab use all different kinds of tools. I'm gearing this talk today for people who may not be neuroscientists, who are family members, autistic people, you name it. But there, I could do so much jargon on this slide. We've got electroencephalography, positron emission tomography, functional near-infrared spectroscopy, eye tracking, all these different and powerful tools for me to harness to try to understand the biology of autism. But when I go into the clinic and I'm a clinician and I'm trying to diagnose autism, or I'm trying to figure out what interventions might be helpful or what interventions might work, I literally have the same tools that Leo Kanner and others used in the 1940s. Right? To engage, to ask, to look, to talk, to play, to listen to parents, to listen to others, and really, it's completely dissociated from the biology. At least in terms of things that we can't see. And I think that we could do a much better job if we could use some of that information in the context of clinical care. And I think the way to do it is with biomarkers. So what is a biomarker? I said I want to steer clear of jargon, but I'm about to show you a really jargony definition that's provided by the US Food and Drug Administration, who actually has a division that works on developing and qualifying biomarkers. And they define a biomarker as a defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. It's a lot of words. But if you really parse those words and think about what they mean, really what it's saying is it's just something that you measure that is objective and biological and relevant to the way a person develops in the context of a condition like autism or without. And really, to think about what a biomarker is, I think it's actually quite simple. If we think of fever, for example, if my daughter has a fever and I put my hand on her forehead to see if she's warm, that's not a biomarker. But if I just use a thermometer, that's a biomarker. And really, that analogy is as simple as it gets, but as powerful as it gets. We don't have any thermometers presently for measuring the biology of autism. And I think that that is a really, really important goal. What kind of objectives would it advance? What would be the uses for biomarkers in autism? There are a range. We might have biomarkers that tell us whether autism is there or not. We would call that a diagnostic biomarker. There have been extremely exciting advancements in the past year or two in this area with tools that use the way a person looks at things that might help clinicians diagnose autism. We can also think even earlier in life, perhaps before autism has manifest, whether there's a likelihood of developing autism. We would call that a screening biomarker. We might think about biomarkers that tell us something about the developmental course a person will experience. Is this going to be a person for whom the autism is experienced as a source of strength? Or is this going to be a person for whom the autism is delivering challenges that that person doesn't want to reckon with? And we might call that a prognostic biomarker. If a person is in the context of treatment, we might think about different biomarkers that could help us guide ourselves in picking a treatment and understanding whether treatment is going to work. We could call that a predictive biomarker. When a person is actually responding to a treatment, if we wanted to see whether that treatment is doing its job, engaging the right systems or showing changes, we could think about a treatment response biomarker. There's one more category of biomarker that I think of as being maybe the lowest hanging fruit for us today in this very, very long journey that we as a field face in getting biomarkers for autism, and that is subgrouping. And the jargon we would use there would be stratification, the stratification biomarker. And the idea is that if we think about this really diverse group of people that we call on the autism spectrum, maybe something that we can measure in their biology could help us split them into more similar subgroups that could be useful for any of the purposes I've described above. And this, I think, is a really important goal, and the one that maybe is closest to being in hand for us in this field. It's also, to me, a striking example of where we have failed to meet certain standards as clinicians. So if we think this is something we already did as clinicians, right? In the DSM, in the previous version of the diagnostic textbook, we had subtypes of autism. Someone wrote in a question in advance asking about Asperger's syndrome. Well, Asperger's syndrome was one of the diagnoses that psychologists could assign before 2013. But after 2013, that diagnosis was taken out of the diagnostic textbook. And those people didn't go away, but the subjective categorical classifications that clinicians had created, were deemed not to hold up, and so that changed. But what a concrete example of how perhaps if we were using biology, we might come to subgroupings that would be more robust, less subjective, and more consistent across clinicians. When we think about biomarkers, what would make a biomarker worthwhile or useful? I think there are some general things that have to be true for any biomarker. The first is what we would call that it's replicable. And that just means it can be repeated, reproduced, and give the same results in different labs. The idea that if I try to measure something that I think is true of people with autism, that someone in a different lab somewhere else using a different lab setup would find the same kinds of things, that those results would replicate. We also want biomarkers that are reliable. So if replicability refers to kind of consistent results across labs, what reliability refers to is consistent results within a person over time. The idea that we're measuring something that is really true of a person in an enduring way and not something that is just true today. For example, my mood is not going to be a reliable indicator of me, right? It's going to change from day to day, whereas my height is going to be something that's really consistent and reliable for me over time. We would want biomarkers that are tolerable for autistic people and children, right? That's just, if not, it's a non-starter. If we want to use these kinds of biomarkers to improve clinical care, they have to be viable in autistic people and viable in children. If we think about helping people at a grand scale, we want biomarkers that are cost effective. The likelihood that we could deploy a biomarker in clinical settings at large scale is going to be at odds with how much it costs to do. That is just a truth of the healthcare delivery system. And then a last kind of basic property of a biomarker would be that the way that we measure it is accessible. Like, for example, if there was a tool that could be used in autism, but could only be collected at an elite autism center of excellence, these blue areas are the people that it could reach. Conversely, if we had a tool that could be administered in any hospital, well, then these blue areas are the people that it could reach. And this is what we want, right? We want things that are accessible. So those are really general, big picture considerations for biomarkers. But what about specific to autism? Like, what are the properties of a biomarker that I think would be most important in autism? Well, really, a basic one would be that we see different results for the biomarker. It looks different in people who have autism and people who don't have autism. A second thing might be, I've talked a lot about social communication, is that it relates to social communication. That maybe even among autistic people, that higher levels of social communication are manifest in the biomarker in one way and lower levels in a different way. And we might also even want to know how specific those relationships are to that domain. And I'll give you that's a little more nuanced of a point. I'll give you an example in the context of a study we've done in a few minutes. We might want biomarkers that could be used in both children and adults. It's not a rule, but autism affects a person throughout the lifespan. And so if we were using biomarkers for these purposes, it might be convenient if there were ones that we could be measuring in children and adults. And then depending on the biomarker purpose, we might want ones that show us changes in clinical improvement. As a person gets better or has difficulties, do these biomarkers vary? Now, this isn't an exhaustive set of things that a biomarker could be expected to do, but I think of some of these as most critical. One of the things that I think has been very, very challenging for biomarker research in autism is to recognize that a biomarker doesn't have to do all of these things to effectively serve a purpose. So if I look at all these things, not every one of these attributes has to be there in a biomarker for that biomarker to serve a purpose. And in fact, if we think about it in a really thoughtful way, you might actually want different attributes depending on how you would seek to use that biomarker. And let me give you two examples. So if I were studying something that I thought might be useful as a diagnostic biomarker, well then it would be critically important that it differentiates autistic and non-autistic people, right? That's the point. But whether it tracks within the autism diagnosis changes in functioning in response to treatment, that might be less important, right? So sensitivity to improvement might be less critical. Conversely, if we were interested in a biomarker that we were going to use to determine response to a treatment, we might have the exact opposite set of priorities. Now, what we really want is sensitivity to clinical improvement, but we might care less whether it maps perfectly onto who has autism and who doesn't. So it's really important for us to be thoughtful about the kind of data that we collect and how we interpret it when we're figuring out how biomarkers might be used to improve clinical care in autism. I want to talk a little bit about a particular kind of biomarker, and this is the kind that I do a lot of work with in my lab. These biomarkers are collected through electroencephalography, which we call EEG. We can abbreviate it EEG. And what this means is it's a way of measuring brain activity, and it's the brain activity that is produced in electrical form from the brain. Many different ways that we can measure what the brain is doing. One of the things that the brain does when the cells of the brain, the neurons fire, they release a small amount of electricity. And if we actually put this kind of net that you see on a person's head, we can actually measure their electricity and see differences in the way the person is responding to different things in the world. Now, what's really nice about EEG is that it's a really user-friendly way of measuring brain activity, right? Really, all you have to do is wear this hat. And for that reason, we can use it across a lifespan, even in infants, as pictured here. We can use it in people who have really wide variation in intellectual ability. It's non-invasive. It doesn't require cutting or scraping. It tolerates if a person moves around. And so really, it's a technology that works in pediatric populations and in populations with special needs. It's also a really practical technology. Getting EEG is pretty inexpensive compared to other technologies. It's really the cost of these nets and salt water that we use to moisten them, and it's accessible. EEG is a technology that we already use in hospitals worldwide for screening to see if whether newborns can hear okay. We use it to screen for seizures, and so it's a technology that's out there. And if we learn something useful in autism, it could be deployed at a very grand scale, very easily and with moderate cost. What's also nice with EEG is that it's been useful in understanding social communication, which, as I described, is something that I think is really maybe very central to developing tools to help autistic people. I want to show you an example of the kind of data that we look at from an EEG. So this is, I'm going to explain all of this, but what you're looking at here is a pattern of electrical activity taken from a person's scalp, kind of over here, over the right back side of their scalp. And what you're looking at is, as the line goes up and down, that's the strength of the brain activity. And it's measured in volts because it's electrical activity. And they can be positive going up or negative going down. What you're looking at here on this horizontal axis is time. And so you see that the entire axis is 700 milliseconds. Now that's 700 thousandths of a second. So this entire chart is less than half a second. It's really fast. And what I want to draw your attention to is this spike that's highlighted in purple. And this is something that we call an N170. The N means that it's a negative pattern of electricity, and the 170 means that it's happening at around 170 milliseconds. So again, very fast, less than 0.2 of a second. And this we've known since the late 1990s is what we see when the brain is recognizing the face, a face. And faces are a really important source of information about social information, and they're also an area in which we see differences often in people with autism, the way they engage in faces. And so a while ago now, we did the first study of this N170 in autism, and we had a finding that there was a really important difference in the autistic people that we worked with. In these adolescents and adults, we found that they showed an N170, but that it was significantly slower than we saw in people who didn't have autism. And this, we thought this could be an important difference because faces are such an important aspect of social communication from so early in life. Now, as I go through a series of studies that we've done, I'm going to keep referring back to that kind of list of considerations I had for autism biomarkers. And so what we've seen here is that this biomarker is different in autism and people who don't have autism. We also wondered then, well, does this have to do with some of the kinds of things that people with autism might have difficulty with? For example, face processing. This is telling us the brain, something the brain is doing related to faces. And so what we did was we saw how good people are at face recognition. And what we found is in this group of adolescents and adults with autism, that they're, despite being just as intelligent as the people who didn't have autism, they made a lot more mistakes on these standardized psychological tests of face recognition. And we saw that how well a person did on these tests was actually related to how fast this N170 brain response was. And so we inferred from this, again, relevant to our list of potential attributes, that we're seeing that the N170 seems to be related to social communication in this way. We next did a study to answer two questions, and this is actually the study that was supported earlier in my career by the Brain and Behavior Research Foundation. Well, one, can we be certain that the differences that we see are really telling us about social information? Or perhaps maybe the brain processes everything a little bit slower in autism. And second, would we see these differences in younger children, the differences that we had observed in adolescents and adults with autism? So to come at this, we wanted to kind of compare something that we thought they were having trouble with, faces, to something that we thought they'd be very good at. And for the thing they'd be very good at, we use letters, because most people with autism, most studies have shown that they're actually really strong, as strong as you would expect based on intellectual ability, at recognizing letters, reading, even if there's sometimes difficulties with comprehension. So we did two things. One is we kind of did the same experiment we'd been doing to see if we would see again, if we would replicate in these younger children the behavioral and brain differences that we'd seen for faces. But then we thought, okay, well, let's look at something totally different that we don't think is an area of vulnerability, we think is an area of strength. And let's look at letters, and let's look at behavior and brain response there. And I'll tell you about both of the components of this study. First, in terms of looking at the social things, but looking at it in younger children, we saw the exact same patterns of results that we had seen in that first study. We saw that the autistic people had more trouble with face recognition, and we also saw that their brain response, their N170 to faces, was slower. So this was important information. It's telling us that we've now, one, replicated a finding, which is a challenge in such a heterogeneous condition, and two, we've seen that it seems to be true across development. You know, from people as young as 6 to people as old as 42, we're seeing the same difference in brain activity. So we thought that was very important. The second part of this study was then to look at the non-social information, these letters. And there we saw a really, really different picture of results. There we saw that there was no difficulty with letter recognition or recognizing parts of words or words the way they we'd seen for faces. The autistic people scored just as well as the non-autistic people and in the average range. And when we looked at their brain responses, we didn't see differences between the autistic people and the non-autistic people. We saw that everybody was responding to letters as they should, with a stronger response to letters compared to these made-up shapes that we also showed them. And there weren't differences in speed, that everybody's brain response was equivalently quick. And so this was important. It was telling us that these differences that we're seeing in this brain system that is related to faces and seems to be a little slower in autism isn't just generically slow. When things are working well in these autistic people, they're working as well as they are for anybody else. The difference we're seeing here is in this particular domain, there's a slowing and there's difficulties with behavior. The last study that we did within our lab to kind of try to understand more about how this biomarker could be useful was to understand how it related to clinical improvement in children who are undergoing treatment. And we were opportunistic here. A colleague here at the Child Studies Center, Pam Ventola, was doing a study of a behavioral treatment called pivotal response treatment. And pivotal response treatment, or PRT, is a naturalistic behavioral intervention that basically consists of playing with kids and teaching them the right kinds of things to attend to get the most out of social interactions and to enjoy them. And in this study that she was doing, these kids came in over the course of 14 weeks for these play therapy sessions. They were preschool age kids. And what we did is we measured their N170 brain response before and after treatment to see how treatment would change things. And what we found is as these kids got better, got more interested in social interaction, more comfortable, more competent in these social interactions, we saw that their N170 actually got faster. And if you look at this chart, you can see on the left is before treatment, on the right is after treatment, and each one of these lines represents one of these preschool children. The dotted line is the average, and you can see how they almost all slope downward, and that's telling us that before to after treatment, they got faster. This is also really interesting and potentially important information that this biomarker seems to change as people change in the context of treatment, specifically with respect to social communication. So if we think about the different kinds of studies that I've talked about and we map them back to these kind of autism-specific considerations that I raised, let's see what we learned about the N170. Well, we saw that it's different in autistic and non-autistic people. These differences relate to face recognition, which we think is an important aspect of social communication. This association seems to be specific and meaningful in that it's not like other things, like letters are slowed down. It's really telling us something that seems to be specific to faces. We're seeing that it's true across a wide age range in children and adults. And in this, I should have qualified as preliminary because it's just a small sample, but in this opportunistic sample of preschoolers, we're seeing that it's sensitive to clinical improvement. So this is where we stood around 2015. And this is, where many research studies for biomarkers in autism stand. A set of small studies done in an individual lab that provide a compelling story, but don't provide a conclusion. And so how do we get to a conclusion? How do we get to a place where we may be able to qualify some of these biomarkers with the FDA? Well, the strategy is as follows. One, you take the biomarkers that are showing the best evidence from these small studies. You then evaluate them in large groups of children so that we can have confidence in the results. And also, the more children that we study, the more opportunity we have to understand the way individual differences among those children might map on to differences in those biomarkers. We use a design that involves different research centers so that we're not worried that these results could be particular to any laboratory, that they're consistent. We use multiple time points, meaning that we measure a person multiple times so that we understand whether it is something that is true of a person over time. And then we use really stringent scientific standards so that we can have confidence in our results, and so that we can think very carefully about, anytime you do an experiment, all the choices that you make are going to affect the outcome, and we can think very, very carefully with great scrutiny about how these kinds of decisions might be affecting results. And then lastly, in case the study works, we want to use the kinds of biomarker tools that would be inexpensive and would be accessible, right, deployable. And all of these kinds of considerations were the building blocks, if you will, for this consortium that we started in 2015 called the Autism Biomarkers Consortium for Clinical Trials. Really specifically designed to take these biomarkers to the next step and closer to actually being used in clinical practice. So the way this study was designed, there's been two phases. The first phase started in 2015 and lasted through 2020. During that phase, we saw 399 children, so 280 autistic children and 119 non-autistic children, which is a really large sample for a neuroscience study in autism. We saw them over multiple time points. We saw them at a baseline, and then 6 weeks later, and then 24 weeks later, so we could see how these things changed or were stable over time. We obtained funding to continue the study in 2020, and we're ongoing now through the next couple of years. This renewal involves 3 different studies. The first study is to bring back those 399 kids from the first phase to see the stability of the biomarkers over the very long term, over 3 to 4 years. The second part of this second phase is to confirm results in a second sample, a replication sample of 200 different autistic children and 200 different children without autism. And then the third part, which will begin, we're in the middle of that confirmation study now. And the third part will be to evaluate feasibility in younger children to see whether the battery that we've tested in the study could work in children who are in preschool. The way we designed the study, what we actually measured is we used really all of the many clinician and caregiver instruments that represent the state of the art in clinical practice. So all these different kinds of things that I, as a psychologist, might administer to make a diagnosis, to estimate progress, and the kinds of things that I would ask a parent to complete or a caregiver to complete to help me understand how a child is doing. In terms of biomarkers, we used 4 different, I haven't talked about them all today, 4 different EEG and 5 different eye tracking. And eye tracking, again, I'm not going to go into detail about it today in terms of time, but eye tracking is just a tool that lets you measure what a person looks at and use that as a way of quantifying biology in autism. And this N170 that we've talked about today was one of the biomarkers that we included in this study. And I want to I tell you a little bit about what we learned additionally about the N170 in the context of this study. Well, one thing that we saw that's really important, and one of the basic things I said was so important about biomarker, is replicability. So this finding that we had seen in all these smaller studies in my lab of these longer N170 timing in autism was true in this really big and very rigorous study. We saw that the autistic children showed longer N170 responses, as we would have predicted. And we also saw that this was very stable across groups. So when we looked at all the different timelines, that when we first saw them at their baseline, we saw them again 6 weeks later, to when we saw them again 24 weeks later, to when we saw them again 4 years later, even in kids, ranging from 6 to then 16 years old across the whole range of ages, we're seeing that this difference in autistic people having a slower response to faces was true. And this chart just shows you the distribution. If you look at all those people that we saw at their baseline, you can see that there's this group of autistic children that have these longer responses, these slower responses, in a way that some of the typically developing children don't. So that was replication, which is really, really important. But we also learned other things too. Well, I guess the second thing I'm going to say is another replication. We saw, again, as we've seen before, this relationship between the N170 and your social function, again, face memory, in that the faster your N170 is, the better you are at recognizing faces. And a new thing that we learned is that this is something that is true, not just in these group differences that I've described, but within a child. So we use a measure, when I say ICC, it stands for interclass correlation coefficient, and it's just how well a person's current values are predictive of their future values or associated with their future values. And we saw this was pretty strong across both groups in the short term and in the longer term, like 6 weeks to 6 months. And so what's so important about this is that we took these findings from kind of a set of smaller studies, made a very, very clear set of predictions about what we should see if this is true of autism in this broader study, and those hypotheses bore out. Now, this is very, very strong evidence. And so what this evidence let us do is then take these biomarkers to the next level. And so the next level would be biomarker qualification. And so the way this proceeds is you basically approach the FDA, you submit something called a letter of intent, and you send that to the biomarker qualification program. And what that contains is the data that you've collected that supports this biomarker as being useful, and that tells the FDA how you think it could be useful. And so we submitted this letter of intent. In 2019, it was accepted for this N170. And as context, even though we've got a long, long way to go before that means it can go into clinical practice, this is the first time that a biomarker for any psychiatric condition made it to this level of endorsement or acceptance by the FDA. And what we said in this consortium was that what we think it could be useful for is for subgrouping, or as I said before, stratification. The idea that when we look at this distribution, and there is that group of kids that I described before, whose values really don't overlap with the typically developing children, and whose values really look different also from most of the other autistic children, maybe this can tell us something about biology. Maybe this is a biological subgroup. And maybe by recognizing the subgroup, we can improve our ability to understand autism. Now, I list here one specific example is even if in the context of treatment trials or clinical trials, we could identify this subgroup, it would dramatically improve our ability to understand the potential response to treatments because we're focusing on this particular biology. And this is a long process. We've engaged the FDA in conversations about doing what's called a biomarker qualification plan over time. It's very exciting work because there's no roadmap. The kinds of challenges that we must reckon with now are really when we have this idea of defining this subgroup based on this biomarker, where does one draw the line? We have to define a threshold. How do you decide where this value is fast enough and this value is slow enough? How do you then validate that? How could we show, because this EEG is this measure of kind of functional biology, are there things that we could show using other tools or using clinical measures that corroborate this idea of the subgroup being something meaningful? And then how do we measure what it means for treatment? The consortium I described, we didn't administer a treatment. And so how could we test whether this subgroup is telling us something different that's relevant to treatment? And then lastly, how can we be sure that this technique that we've used among this really expert group of research labs could be rolled out in a broader way at other sites? And so these are some of the questions that we continue to wrangle with. It's hard work. It's scientifically challenging, but I think it's immensely valuable. I mean, when we think about autism, a condition that I care so much about, it wasn't even talked about until the 1940s. And now here we are at a stage where we have biomarkers under consideration where many other conditions don't. And so I think that it's exciting to have reached this point. I want to talk now about two other lines of research in my lab that are trying to leverage this biomarker science to develop new treatments, and then also trying to include new groups in this biomarker science. And so one line of research is trying to understand whether this N170 could actually give us a treatment target. So we know that the N170 is related to the function of a specific multiple parts of the brain, but one in particular called the superior temporal sulcus, or the STS. And this is a part of the brain that's really important to social perception and to thinking about others. We also know that from other kinds of brain studies that when people receive treatment and become more comfortable in social interactions, this is the part of the brain that we see looking different. And so the question that we're exploring now is, could we use this N170 response as a guide to boost function in the brain? And to do so, would that give us the opportunity to enhance social communication social communication in autistic people who seek to have greater comfort in that domain? Now, the way we would do that is with this tool. This paddle is called a transcranial magnetic stimulator. And what it does is it actually releases a magnetic current that can increase the activity in a particular brain region. And so what we're in the process of doing and learning about is actually seeing whether targeting this brain region where we see this N170 and it's slower in autism, whether ramping that up would actually improve comfort in social contexts. We're in the process of doing this work in autism. We've got very exciting proof of concept in people who don't have autism. And what we find is that when we stimulate this part of the brain, we see, even in people who don't have autism, their N170s get faster. So you can see before treatment and after treatment, their N170 has gotten faster. And so that's the direction we would expect to see it move if it were to be useful in autism. And so this is ongoing work. Stay tuned. The study is in progress, and we should have results within the year. The second line of research that I want to mention is really highlighting what I see as a major problem in a lot of the work that we've done, and really truly in most neuroscience research. And it's relevant to biomarker research because much biomarker research is neuroscience research. And here is that most studies don't include people with autism who also have intellectual disability. Like in that ABCCT study that I talked about, we're only including people with an IQ above 60. But 38% of people with autism have an IQ below that range. And we recently looked at literature and we saw that only 1% of the neuroscience studies that have been published have included this population in them. So this is, for me, a really glaring omission. The idea that that we have failed to include the people who could potentially most benefit from neuroscience research in the neuroscience studies. So what can we do? Well, our strategy, and I've shown here Adam Naples, a colleague here at Yale, who's really led the technology associated with this work, is trying to develop new tools that make it more feasible for inclusion of autistic people who also have intellectual disability. We've done three things. We've changed the way our laboratory is physically set up and the tools in it. We've developed with the collaboration of a behavior specialist, a very specific behavior protocol, and we've changed the way we administer experiments. I want to focus on the lab setup. What Adam has done is basically set up a system in which the laboratory itself monitors with cameras how much a person is moving and whether they're paying attention to the screen. And what we can do is then set up a contingency wherein a person gets to watch their favorite videos if they sit still. And so a person might come into the lab, they're moving around and watching their video. At some point, they're naturally going to sit a little bit more still, and we can pause the video when they do that and adjust the threshold of movements that's tolerated, and basically teach a person without interfering, without using words, that sitting still in this room is play the video. And we can then use that context to show things that are a little bit less interesting than videos, but are kinds of our EEG experiments. And what we find there is that it really works. So this chart is a little hard to understand, but what I want you to see is whenever you see blue here, this is an experiment, is a person's not paying attention. And whenever you see one of these lollipops, it means that we're able to run part of an experiment. And this is all done automatically. And so you can see when a person's not paying attention, the system learns and it stops showing the experiments until the person re-engages. And our hope is that this system will let people with autism and intellectual disability participate in ways that are kind of meeting them where they're at. And so far, it's really working. In the data that I'll show you, we've had a great deal of success getting this to work with people in a group whose average IQ is 31, which is in the range that we would describe as severe intellectual disability. We see that in these people, the response patterns we see seem to be working. In other words, that we're seeing the stronger response to faces at the N170 that we would expect. And we're also seeing some of the same kinds of differences. So what you see here is that the speed of the N170, these are people who don't have autism. You can see what we'd expect, like from the ABCCT, that the people with autism without intellectual disability are slower. And we can see the same pattern, even more pronounced in those with autism and intellectual disability. And so in the context of a field that's recognizing that those with severe autism, those with profound autism have been excluded from much of the research, we're very excited about this approach so that we can hopefully maintain a much more inclusive stance in our research program. I want to wrap up. I want to thank a few groups of people. Most importantly, the autistic people, kids, the adults, and their families that come in and work with us. Autism science is a partnership. We can have the tools and we can have the scientific knowledge, but we can't learn anything unless autistic people and their families are invested in working with us. And so we are eternally mindful of that and eternally grateful of that. Even though we're scientists, we recognize that this relationship comes from our clinic. And so I want to acknowledge the clinicians who work so hard every day to help autistic people and their families in the Yale Developmental Disabilities Clinic. I want to mention the broad and distributed group of people who contribute to the Autism Biomarkers Consortium for clinical trials around this country, and then our collaborators internationally. And then lastly, I want to mention the group of people right here in New Haven in our laboratory who are doing so much of the work that I've had the pleasure of describing to you today. I want to, before we go to question and answer, I want to show one more slide, and I want to thank one more group of people, and that group of people is you. I want to thank you because I want you to consider helping us do this science, and please let others know about the opportunity to get involved in research. In the studies that we have going on in the lab, like the Autism Biomarkers Consortium and others, we're working with people aged 3 through adulthood, people who have autism or don't, or people who have intellectual disability or don't. We will try to make it worth your while. Every person who comes in and works with us has a very thorough clinical evaluation report. We will compensate for your time, and we're respectful of the inconvenience to you. We'll do everything we can to make sure that you are fed and that you have transportation and things like that. So if you are interested in partnering us in research, or if you know someone who might be, please send them our way. Bella Panjevic is our super warm, friendly, and clinically talented outreach person, and this is her email and phone number, and you can learn more about these and other studies at our lab website, www.mcp-lab.org. And thank you for your kind attention. I'm happy to use our remaining time to answer any questions that may have come up during the presentation.
Jim, I want to thank you first and foremost for an outstanding presentation and the work that you've done over the years and continue to do. And it really shows the relationship between basic science, clinical work, the development of potential biomarkers, and potential interventions. Such important work. So thank you for all that you're doing.
Thank you.
I want to start off by asking, what you spoke a little bit about where this is all potentially going to go. What do you see? What are your hopes for all of this work in the next 5 years, next 10 years? How will this actually help people living with autism?
It's a great question, and that's the most important thing. So where we are now is, I didn't talk a lot about kind of international efforts, but both this consortium that I've mentioned and a European consortium called Aims to Trials. We both worked with some of the same biomarkers in the USA, working with the FDA, and in Europe, working with the EMA, the equivalent. We're both at the point where we have gotten these biomarkers under consideration and are collecting evidence both in our own studies and together to try to make the case that these could be trusted and useful. And if we can make that case, well, then they can start being used in clinical trials research. And I think that would have very, very far-reaching advantages in a few ways. One, a problem in autism is that many intervention studies lack sensitive outcome measures. And so there's no way to really be confident for whom a treatment is going to work and to understand why it might be working in some people and not other people. I think this would address that problem. I think also a more practical consideration is that once we have advanced some of these biomarkers, we will make it so much easier for industry to get involved in autism research. It's just when we think about the kinds of research resources dedicated to developing new treatments for psychiatric conditions versus other conditions, they're so much lower in psychiatric conditions. And part of the reason is because it's so hard to measure change. If I had $1 billion to invest in research and development for a treatment, I don't know that I would choose to invest it in something where a play session with me is going to tell me whether that was a good investment. And with these biomarkers, we'll be able to, I hope, get really a qualitatively different level of investment in developing new treatments.
Good, excellent. Let me ask you, what's the youngest age that you envision this type of biomarker as being helpful in terms of the important issue of early diagnosis?
That's a very important question. The work that I talked about today is, we're studying this in older children. So the research that I've described is really tangentially relevant to that. I mentioned earlier that there really is very promising work from other groups at Duke, at Emory, and in corporations using a technology basically where the camera of an iPad or similar tablets or other proprietary devices can be used to measure how a person looks on screen. And what we're learning from that research is that very, very early in life, months into life, there are differences becoming evident that are helping scientists and clinicians to predict who's going to develop autism and who's not going to develop autism. And so really these kinds of tools, maybe not the specific tools that I described today, but these broad biomarker approaches using other related technologies like eye tracking, promise to, hold short-term promise to really change the way clinical practice could work very early in life, which is important because the sooner we know how a person is doing, the sooner we can give them the support that they could benefit from.
Great, thank you. One last question. If somebody is listening to the webinar, a parent, other caregiver, who have concerned about their child, they have not yet received a diagnosis, they haven't been evaluated, what should they do? What do you do when you're concerned? What's the steps that a family should take?
Well, the most important first step would be to talk to their physician or their pediatrician, because that's going to be a great base to get connected to all different kinds of resources. Many pediatricians are going to feel very comfortable speaking to the kind of early features of autism and making a determination of whether this is or isn't autism. But then also they're going to know how to connect families to the kind of really specialized clinics, the kind of clinics that we run here at Yale, where we're going to work with a child from a multidisciplinary perspective with psychology and psychiatry and speech and language, and really going to do a kind of comprehensive deep dive to understand this child, to work with parents to understand developmental history, and really, not when you think about it, the diagnosis of autism is less important than thinking about what is a child having trouble with and how can we help. And so these kinds of really deep, comprehensive assessments are most useful in this regard. But I think always a pediatrician is a great place to start in terms of trying to get needed attention for a child who's experiencing some difficulties.
Thank you. Jim, your passion for this comes across so strongly. Thank you for all that you do. Thank you for joining us today. I want to, in addition to thanking Dr. McPartland, also thank everybody who joined us for the webinar. And I'd ask you to please consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This means that when you donate $1 for research, that dollar goes directly to the scientist. To make a gift, please visit our website, bbrfoundation.org, or call us directly at 1-800-829-8289. This webinar has been recorded. If you've missed any portion or would like to share it, please visit the events and webinar page on our website. And please join us again on March 12th at 2 p.m. Eastern Time, when Dr. Juliet Edgecombe, an assistant professor at the University of California, Los Angeles, will present detection of suicide-related emergencies among children using real-world clinical data. Thank you, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Thank you.